Hi, everybody. My name is Rizwan Chaudhry, and I'm your host for a brand new podcast called Organoids and a Large Portion of Chips, which is being brought to you by WORC.Community. And we will be talking about all things organoids and organ on a chip. And I'm delighted to be joined by my two regular and resident experts, Professor Mariana Krotov-Julia, who is head of Urology Research Laboratory at the Department of Urology and Department of Biomedical Research, Director Translational even Organoid Research Resource. I'll get my teeth in a minute. An associate member NCCR RNA and Disease at the University of Bern, and thankfully with a much shorter job title, Professor Stephen Ray Wilson from the Department of Chemistry at the University of Oslo. So first of all, welcome Mariana, Stephen. I'm delighted to be hosting this podcast with you. How are you both?
I'm doing great. Really looking forward to this podcast. I like organoids. I like chips. I also like chips that are not organ chips. I like Doritos, Pringles, everything. So that's my half American part of me that is really into this. So this is perfect for me.
And before Mariana comes in, I have to point out to you that chips in the US is very different to chips in the UK, because chips in the UK are fat French fries in the UK. So, and we would call them crisps when you talk about Doritos and things like that in the UK. So it just
Oh,
shows the difference in language. And Mariana, tell us, it's lovely for you to join us. Tell us a bit more about you. How are you?
I'm doing wonderful, and I also like crisps a lot. Maybe not associated with the organoids, I think. Although I really like I'm really an organoid kind of fan, and I like the potentials that this new technology can I think bring to us, and I think it's fun to talk about it. And I think this is a really nice setting for it. And I just realized that my background is the other way around. But okay, but that's okay.
Yeah, actually, so is
oh,
mine. Actually, I've just realised, but that doesn't matter. In fact, yours are right. Mine is wrong, I think, actually. So don't worry about it. So
Interesting.
we'll see how it goes anyway. But look, the wonders of doing a live podcast. So look, now
Yes.
before we talk about organoids, though, and it's lovely to meet you both, and glad that you're well. For viewers though who don't know about your expertise, though, Maria, perhaps you can kick us off first. Though. Can you give viewers a quick overview of your background, the work you're currently doing in this space, and why you think there should even be a podcast on organoids and organs on a chip?
So yes, so my background is on cancer biology. It's a background that I acquired, let's say, in the last 10 years of research. I started out doing something completely different. I studied chemical physics, so I was a completely other background before. But then at one point there was this kind of fascination with the stem cells and the potentials, and then that brought into the study of prostate organogenesis. And then from there, the step was a really quick one from prostate organogenesis to prostate cancer. And prostate cancer has been my line and focus of interest for the past, well, many years by now. And recently we actually expanded also, the lab has expanded to bladder cancer as well. And we've now also in the last 5 years touched upon pancreatic cancer.
Hmm.
And the reason why we've been expanding and looking at it from so many different cancer, from solid cancers, from so many different organs and sites of origin, is because one thing that we realized in the course of the study is that there's many commonalities. And organoids actually are a very interesting way to study, interesting tool to study these commonalities. And maybe, I think it's really nice to bring it to more general public, because a lot of people talk about them, but not a lot of people know what they are. So I think it's really cool to have this podcast to chat about it.
Fantastic. And Stephen,
y-
your background, what work do you do, and why are you fascinated by organoids, and why we should be doing a podcast on the topic?
yeah. Well, I was supposed to be a psychologist, actually. I started, when I was in high school.
I can talk to you after this podcast, and I've got a few questions to ask you. So don't
Absolutely. I would
worry.
love to do that. No, I was I was supposed to be a psychologist, so I started studying psychology at the university, and I thought it was going to be all this stuff, Freudian stuff, and all this, but the stuff that I thought was coolest with psychology was, it was about neurotransmitters and stuff like that. And that actually made me do the jump over to start studying chemistry. So and then my intention was that I was going to be studying chemistry to then do more biology stuff, but then I just kind of got stuck, and I kind of fell in love with chemistry and then
Yeah.
analytical chemistry. So I've been working for many years with analytical chemistry, which is basically measuring stuff in stuff. So that can be I used to work for the police, like the narcotics lab, to see, what kind of drugs people were taking, right? And I've also been in a lot of other applications, but the organoid stuff has just been a perfect mix for me because it's very multidisciplinary,
Right.
very challenging samples, and it really there's a lot of new stuff to grab on, and it's just such an exciting and really emerging field. But listen, Maria, I have to ask you, you started with physical chemistry,
Yeah,
was that?
I was studying the Schroding equation.
Oh man,
Hardcore.
that's some hardcore stuff. But how did you what did you think? I mean, that's kind of like a leap from psychology to chemistry, almost going from Schrodinger to organoids. Was that tough for you, or how that work out?
Well, I don't know. I think I was young, I guess, and I just wanted to I got caught in, these European networks that we still have, kind of the Marie Curie ITN networks. And the person where I was doing, so the PI where I was doing my internship was
Hmm.
part of these networks, and it was like a bit of a weird thing because it was a combination of really hardcore chemical physics with biology. And what happened was that I ended up going to one of these meetings, and the meeting was in Cambridge, actually, in the UK. And one of these people gave this lecture, and they started talking about stem cells and kind of basic. And I was just so fascinated by it, that I thought that I wanted to to know more about it. And so what I then decided to do was then I wanted to do a PhD with one of these people in the network, which I didn't end up doing, by the way, but I ended up instead doing a PhD in the Netherlands at the medical faculty, so in at the AMC, and there I focused mostly on liver regeneration. And that was just, a really, really fascinating way of starting. And then from there, the leap to stem cells was a very little one. But I guess my friends say that I have like the rubric syndrome, so I gotta solve the puzzle kind of thing, you know. So, so and I think that uh applied biology is really something that fascinates me, and in particular in the last years, translational medicine. And I think this is a good time to call back the word organoid into this conversation, because I think the organoids have a huge potential in this translational uh, medicine that I don't think we have managed to really unveil yet. And of course, the organs on chip also are extremely an important aspect of this, I
Uh,
think.
well, Mariana, that's a beautiful segue actually into my next question, which
Okay.
was for those viewers who are, or listeners, I keep saying viewers, sorry about that, but the listeners here to this podcast, but those people are going, well, this all sounds very exciting, but what the hell are organoids and what are organ on a chip? Perhaps one of you would be kind enough to actually explain what organoids are, first of all,
Mhm.
so they actually understand what we're actually talking about, and also what organ on chips are, how they differ.
So if I may, I think that So I think that the common knowledge of what an organoid is like we take a little piece of tissue from a human that doesn't have to be affected by any disease, or it can be affected by a disease. And the idea is to figure out a way to grow this tissue in a dish, and form three-dimensional structures to it, which still sounds quite a very complicated way of describing this. But originally, I think that the organoids started by looking at stem cells. So by looking at cells that can form a tissue by themselves, or organs by themselves. And there's a huge literature, of course, that started out in the Netherlands with the LGR5 and the colon, and probably everybody knows about the backgrounds with the Hans Clevers and the pioneering of this with the colon. And I think from there on, everybody has found their version of either the stem cell or a different way of studying this by forming these mini structures in a dish. I do, however, want to point out that the organoids were not discovered by Hans Kleber. So the concept of the organoid, of course, is something that actually I was very fascinated, because I looked up some literature, and I think the first cultures are really the spheroids, they call them, were back in the mid-50s even. So where they were talking about cell aggregates and things. So I think if we start digging back, what we do is that we basically kind of repeat and improve history in a bit of in a different way. And yeah.
So are organoids then, are you trying to replicate a specific organ then in the human body? Or is it just when you say organ,
Yeah.
are you trying to replicate the colon or the liver or the pancreas or whatever? Or is it just an organ, but it's sort of ambiguous what that organ is?
So it really depends on where the tissue, or what you start out with, the cell you start out with, right? So if you start out with a cell that is a liver derived cell, maybe you're trying to make something that looks like a liver. If you start from a pancreas, you start with a pancreas kind. And the fascinating thing is that, as I said, the cell that you have doesn't have to be diseased there, but you can also start from a diseased kind of cell. And
Right.
then what you're trying to do is that you try to recapitulate the disease itself. And that's where the word translational medicine comes mostly into place, in relation, for example, to oncology, because that's I would say, uh, the largest application we have for the organoids. Not necessarily the one that works the best, but at least the one where a lot of people are certainly looking into it. So it's
Cool. Stephen, do you want to ask anything? Because I've got one more question from around, talk more specifically about organ on chips. But before we
No,
move
go
that,
for it. Absolutely.
so then what is an organ on a chip then? And how does that differ from working on organoids?
So the organ on a chip is a much more sophisticated and complex system, I would say. It's an organ on a chip is something that um, represents the multitude of different kinds of cells that are present in the body. I mean, you are made of so many different kinds of cells. It's not that if I take one cell of you and I put it in a dish, there I have you in my dish. That's really not the case, right? And for the organs, it's the same thing. So you have blood flow, you have immune cells, you have muscle cells, you have all different kinds of cells that participate, that go under tension, that interact in a certain way, not only at a 2D level, but also in a three-dimensional level. And that's the power of an organ on chip kind of system, a power that an organoid system instead does not have yet.
Right.
Say, yeah.
And so I'm going to bring you in here because obviously you are involved very much in measuring things and analyzing things. So when we start talking about organoids and organ on a chip, how do you analyze those sort of samples in comparison to what traditionally you may have sampled, maybe small molecule or whatever?
Yeah, sure. Well, Well, first I always have to go back to the 90s and watch Jurassic Park again. That's kind of what, that I mean that's kind of the vibe you get from these organs that that you can grow these amazing, replicas. I mean the imagination and the intuition of it all it can be very exciting. And of course there are limitations though with organoids, but they're consistently evolving. So my job a lot of the time is that I work with organoids, and I have very close collaborators with different, developmental biologists and medical researchers, etc. And I'm then provided with organoid and organoid chip samples, and then I typically do measurements with something called mass spectrometry. So that's one of many common analytical techniques. So if you hear about people caught in doping, in sports, or if they found some pollutants in the ocean or whatever, then it's a pretty good chance that it's mass spectrometry
Sure.
that's that. And a mass spectrometer is it's like a really expensive weight, like a balance, so that but instead of weighing people or whatever, then it can weigh individual molecules and give a signature of what these molecules are. So I use mass spectrometry and other techniques then to measure drugs and drug metabolism, which might tell something about how a liver is, handling a drug, or maybe we can measure like the proteins or the lipids in the organoids. And this way you can compare organoids from healthy to diseased organoids, between organoids, and comparing them also to living patients and so forth. So being able to measure stuff is really important to see how something is doing, like for example, organs and organoid chip systems. And that's where I come in. And I really like doing, because these are, even though they're tiny samples often, but they're very complex, compared to many more simpler samples, obviously, and you want to do things in a very reproducible way, because this is now a field that has been exploding quite a bit, but now we have to make sure that we're going to get reproducible results, and we can use this for stuff, so that this continues to be a very promising tool and a very much used tool.
And it's
Yeah.
amazing actually how you two are so good at giving me the next bit of my question, because that's actually a perfect segue to what I was going to ask next, which is we're here talking about organoids. We're doing this podcast all about organoids and organoidship. And from my perspective, it's become more prevalent in terms of what I'm seeing in terms of the press and the media over the last 12 to 18 months. I've seen a lot more papers, news stories, and so on, and also events, let's not forget, around organoids. So, what do you think has driven that explosion or increase of awareness of organoids and organoidship? What do you think has driven that to come to the so that it's come to the fore now, as opposed to say 5 years ago, whatever? I mean, see, maybe you want to kick us off on that from your point of view, from an analytical point. Then, Anna, maybe you want to talk more from from the research perspective, what's been
Yeah.
helping you drive that?
Well, I think that again, there's something very attractive about these mini organs intuitively with the general public. I mean, you can tell if you tell somebody that you're working with organ models as, for example, a way of not having to do so many animal experiments anymore. I mean, people can kind of intuitively understand that that's something they think is cool in another way than if I tell about mass spectrometry, or if Mariana's going to try to explain the Schrödinger equation, in the elevator. It's tough. Tough. I mean, I also like a little bit of quantum physics, but I'm still confused, therefore, I think that, the public, they can kind of catch more on when it's written properly about it, like in popular science, or even in the newspaper. In Norway, there have been several, major newspapers that have made pieces about organoids,
Really, right? Okay.
and also but again, back to that this can reduce a number of animal experiments. This is also something that that can catch the public's eye, because animals, of course, there's a lot of ethical considerations around that. I mean, I think that it's in the 100 million scale of animals that are sacrificed each year. And we're seeing stronger regulations against it, but also there is a need for alternative models that will represent, parts of human physiology better for the given experiment or test that is taken. So I think, and that's also kind of what's got me into it. And I also see that a lot of students, a lot of young people, at least in our university, it really makes them, their imagination and research and science really come alive. So yeah.
And Mariana, what do you think has sort of driven, I won't use the word explosion, but a greater awareness of organoids and a lot more exposure for organoid content
Well,
and papers?
I think that we had different in the history of science, we have had different hypes of several things, right? I think 20 years ago we had the genomic hype, and we thought that we were going to sequence a genome and cure all diseases that were out there, right? That unfortunately didn't happen. But I would say that in the beginning of let's say the 2010s, 2011, I think the organoid kind of hype kind of started, or the creation of these models. And maybe at one point during this series of podcasts, I think one of the things we should maybe chat about is also the nomenclature of the organoids, meaning that we call them organoids, but really the ones that we relate to cancer are really more like canceroids, I would say, right? So I think there's also a bit of confusion there in the name, which I think is really important, because people get confused, because like you
Sure,
said, I mean, I call them organoids, and then you think that I have an organ in a dish, but that's not necessarily the case.
sure.
But I do think that the real hype has come in the last few years, that came and it comes from the fact that now technology has changed a lot, improved, and we can do a lot of spatial analysis as well, and also spatial transcriptomics. So we can look at the expression of the different genes in these organoids. And the potential of these organoids is that you can do this thing that's called a longitudinal measurement, meaning that you put a drug on these organoids today, and then you can analyze it in time and see how this has changed the landscape of the organoid, of the transcriptomics, or the gene expression in there. And that's actually really powerful, because that means that, imagine now, for example, related to a disease like cancer, right? The disease like cancer is a disease in which, like many disease, it starts in a way and it progresses in in different other ways, right? And the question is, how does it progress? Can we figure that out, right? And the idea of having in a dish something that we could try to mimic this progression means that maybe, who knows, at one point we might be able to predict and prevent the course of this disease just by studying it in a model that is, as Steve said, not related to an animal model, but related only to the human, which potentially, especially in this area for the 3RC reduction, is huge, I would say. So yeah, and if we could couple that to organs on chip, well, I think, it would be really, really a nice way of of studying the diseases, the biology, and also the course of the disease, I think. Yeah.
And how important do you think the use of AI has been in driving that? Because obviously, I mean, you talked about mass spec, Stephen. As I said, I have the pleasure of attending the ASMS conference every year, and I see the mass spectrometers get more and more powerful. Every time I go, they can do far more detailed, exploration of cells and so on, and get vast more data. But I know that's driven a lot by incorporating AI into those instruments. and great software as well to make sure they can do that research. So how is AI driving that, and uh, how comfortable are you using the AI? I mean, I suppose how trustworthy do you think the data is? Because like everything is what you put in is what you get out. And so, and AI is one of those areas, isn't it? People are sort of debating now about the use of it. So how important is AI in terms of driving drug discovery, and particularly in this space?
So for my particular field of let's say the cancer field, I think AI plays a major role right now. I mean, I don't think that any of the things we do anymore can be done without the assistance of AI or developing software that will allow for this multidimensional kind of analysis. It's just not possible. The amount of data we have is too much to process. Of course, I say this also with a grain of salt because you also have to relate what AI predictions are in terms of drug, for example, administration to patients, or drug predictions in terms of potential therapeutics. You always have to counterbalance them with what actually happens to a patient. And to this point, I think that the clinical trials that we run at the University of Bern, in which we match the organoids prediction with the AI software, are going to give us answers on in the near future on how predictable the AI software is and how useful actually it is or it is not for the prediction of drugs to patients.
And Stephen, from your perspective, obviously, as
Yeah.
someone who's using mass spec a lot as well in terms
So
of your analysis.
yeah, I mean, AI, I think is, I mean, I was teaching a class earlier today, and we were just looking at a review paper, and it showed some predictions, and this was like before AI, or at least before AI became mainstream. And it's just funny to see some papers just from some years ago, that how insanely far along we've come now with the AI. But I would say that from an analytical chemistry point of view, which a lot of time is related to quality control and optimization of methods. Like you say, what you put in is what is going to come out. And I think that in like chemical analysis, also for organoids and organoid chip, it's going to be more and more important to have extremely reproducible and standardized assays and measurement systems. And this will then help also the AI to give better information. And I would just like to say, it's not always the most advanced instrument, or the instrument that gives you the most information, that will necessarily be the most hardcore in predicting stuff. And I can give an example. So nuclear magnetic resonance
Yeah.
spectroscopy is, it's an instrument that it's not particularly sensitive compared to mass spectrometry, which can show thousands of peaks. But like NMR, as it's called, it might see like 40 or 50 metabolites at a time, but it's super reproducible, right? I mean, if you do a scan in Australia and then you do it in Norway on the same machine, it'll give you the same result. And this type of input, then you can go nuts and bananas with the AI, and it's shown a massive amount of predictability for diseases and so forth. There was a paper on this in Nature Medicine a couple years ago that just totally blew my mind. So um, I think that from an analytical point, it's important that we keep on are able to identify more compounds and stuff. But I think equally important is that we ensure that we have validated and reproducible data, and that's something I think will also be common for organoids and organoid chip system, is making sure that there's no kind of X factors going on, that why is this happening, and why can I reproduce these results. And I could use a whole podcast series about that. That's my take. What goes in, what goes out, that's going to be an important part, as always, for AI, also in analytical chemistry.
Cool. Right. Now we've done a nice little overview, I think, for listeners to so they understand a bit more about organizership. So let's talk about the current situation then. I mean, you guys obviously are working in this sector, and I understand there's been quite a lot of different news papers, etc., being published over the past few months. So anything stood out for either of you in the last few months in terms of papers which have been published or any research that you've seen? Mariana, maybe you'd like to kick off.
Yeah, so I think there's been really many publications that have gone out in the past months, and um one of the things that I see a lot of them also because I'm editor-in-chief of a journal, so I see also the kind of the background, the ones that
Sure.
don't reach the publication level. And I think that the so the ones that I find particularly interesting that have made a significant step forward, I think, in our field are ones that are also in my field at least, that are related also to bladder cancer.
Right.
And I'm saying this because bladder cancer is actually quite an understudied disease out there. A lot of people have it,
Yeah.
but it's not how can I say, very popular to study it. And I think one of the biggest the problems that we have had or we have with the organoids derived from the bladder cancer is that mm, they cannot be cultured indefinitely. Now, so I would like to make this point because I think there's it's a very important it's a very important concept out there. And I think that one of the things that I am not going to single out any single journal or manuscript that studies on this, but what I would like to say is that I think there has been a real shift in the field in the last, I would say, few years, in which people have now accepted the fact that to have an organoid in a dish, you don't necessarily have to passage it indefinitely for it to be defined as an organoid. And I think this concept has now come out in many different publications that are out there. And the example that I wanted to bring out was the example in my field because, well, it's the, let's say, the field that I read more about in terms of organoids and technology. So now we can culture organoids of bladder cancer and go from a biopsy to an organoid and to a drug screen in just 10 days. So it takes us only 10 days to do this. And what we have is to reassure also Steve, a highly reproducible system.
I'm sure you do.
But not necessarily the organoid is reproducible among different patients. And that I think is what gives the fascinating aspect, because from this, the major thing that we learned was something that we actually already knew, but we didn't have the proven proof of it,
Sure.
is that actually every cancer is highly different among each other. And if you want to study the cancer and if you want to look at therapy, you should not have the organoids adapt to the culture conditions, because you don't want to change what you have in the dish. You want to test as fast as possible so that it stays as close as to the original
Sure.
dish. But with that said, I am not saying that long-term cultures are not good. So that's also good to have, because maybe people have different kinds of questions. And I mean, about the CRISPR-Cas technology, right? So it's people have won Nobel Prizes for this as well. And CRISPR and this particular technology cannot be applied for short-term cultures, but can be applied in long-term cultures. So the scientific question that you're asking is different, but that's okay. Both are okay, and I'm sure we could find ways of measuring metabolites and mass spec information from both of them. Right, Steve?
Absolutely. And I just also want to just comment on that, that you were saying how different people are different, and people's cancers are different. And this is also has such enormous potential for organoids, organoid chip, whatever, is that you can um, make systems that would represent one patient or patient group and another patient group. And you can do this. So I guess, personalized medicine, it's at least it's an expression that kind of gets thrown around a little bit. But
A little bit, a lot,
there's
I would
definitely a
say.
personal touch to this. Absolutely. Hmm.
Right. Okay. And Steve, from your perspective, anything that you've seen in the media or out and about on organized that you'd like to talk about and share?
Yeah, so it was funny. We were talking a little bit about hype earlier today, and there's been a paper that was written by Charles Oliver and co-workers that it's a perspective called "The Roadblocks Confronting Widespread Dissemination and Deployment of Organon Chips," and that's a mouthful. But in the paper, the perspective they're talking about, the fear, the consequences of hype, right? And that sometimes a concept it might be almost a little bit dangerous for a concept to have too much hype, because then people might get overly, oh my God, we're gonna make Frankensteins that can solve everything or something like that, right? And they say, oh, but you're making a liver? You mean it looks just like a human liver? Everything is the same. I mean, the public and maybe even investors and even scientists, they can get a bit carried away with what they are expecting that these things can do. And I think that one of the points in this paper is that we have to keep on communicating, with everybody from the scientists to the big pharma to the CROs and stuff like that, so that we're going to make sure that this is going to be done soberly, properly, and also with a lot of excitement. Again, like proteomics, that also went through this hype circle. What are these things called? There's I think it's the hype circle, isn't it? It's called the Gartner hype cycle. That's what it is. And the Gartner hype cycle, it starts with that there's a lot of initial um, excitement, and then you get up to a peak of inflated expectations, and then it goes down to the throw of disillusionment, and then up the slope of enlightenment. This is kind of like a standard thing we see that it's being applied. And I think like proteomics had that a lot. Marianne was also talking about genomics. People think, oh, we're going to solve everything. And it's, oh no, it didn't solve anything. And then it just kind of like, ugh, this is just, ugh, right?
Yeah.
But then slowly but surely, it kind of builds up. And this paper suggests some procedures for removing these roadblocks, and a lot of this is to have communication between the stakeholders, and I guess trying to keep it real in a bit, and trying to have a good communication, also multidisciplinary communication between the biologists, the engineer, and so forth. I think also like AI is the same in this, because um, AI is definitely going to be a big part of everything, but then we have to also include AI researchers and not just expect an engineer to do this for us, because every field needs hardcore science and researchers in basic research, and that we, respect each other and collaborate. So I thought that was I thought that was kind of a cool paper, a bit provocative, and I think that's good for the field.
Cool. And we'll put the link to that paper actually above this podcast, so you can actually click through. And in fact, it'll all be at the WORC.Community website as well. You'll be able to find any of the papers that Mariana or Stephen mentioned on this podcast. You'll be able to find them there as well. Now, one thing, actually, Mariana, what's your view of what Stephen just said about hype, though? Because I've got another question related to that, but I want you to sort of, what's your feedback to what Stephen said about hype and how you should manage it?
So hypes and how to manage them. I think Steve is very right about this. I mean, I think there's highs and lows of these hypes, and I think it can get dangerous in the well, dangerous is a big word, but I think it can be confusing for laymen, I would say, people, because we as scientists, we talk to each other in this um, language that is very clear to us, right? And because we read papers, and this is what we do every day. So even when it, you know, also when you work on cancer, you kind of dissociate from the concept of the disease. And so, I talk about cancer every day. I think about cancer every day. And but it's not it doesn't affect me in a way. But I do realize that when I talk about it with people that are not in my field, it's like, oh wow, cancer. And I'm like, yeah, but it's just a disease, right? But it's not, right? It's perceived by other people in a very different way.
Sure.
So I think that one of the things that we as scientists should learn is to actually talk to people in a different way. Not necessarily talk down to people. That's not what I mean. But at least talk to
Dispassionate, maybe.
yes, but talk to make sure that people understand what we're trying to say. And also when, one thing that I never do is I do not talk about animal experimentations with people that are not in my field. And the reason why I don't do that is because it's too hard for me to explain why I have to inflict pain to an animal to understand something, right? Because people associate the concept of animal experimentation to their pet dog at home, which of course it's if you relate it like this, it's very different, right? But I think that's also my fault because I don't convey the animal experimentation in the right way. Like in a way, it's difficult for me to convey the organoid experimentation in the same way. So I think it's really a matter of uh, how you talk and how you try to explain things. Like to give you an example, right? I was interviewed for one of the papers that came out. They did a press release and then we were interviewed, and they commented a couple of quotes, right, that I had. And this thing ended up going on the local newspaper, right? But it's like I don't know what version of local newspaper you guys have there, but it's like the Metro. So something that you pick up,
For free on the
yeah,
train or
exactly.
whatever.
So you're there. They open, people open this newspaper, whatever, and they read quotes and stuff like that. And then I started receiving emails by people, or phone calls even, by people asking me if I could cure their cancer. And I was like, oh my God, that's not what I said.
Yeah,
Yeah,
that's right.
that's not what I said. That's not that. And then I realized that I just said it wrong, or at least I conveyed it. What I wanted to say was not what came was received, right?
Yeah, that is,
So yeah. So I think we have a very important role in this, in trying not to um convey information, not misinformation, but information that can be misleading to
Or
people.
distorted, maybe even there's distorted, I would say,
Yeah,
because
distorted.
but then I think your point is true, but obviously, unfortunately, when you do something like that and it gets taken out of context or gets put into a uh, general public media, is going to have a different meaning than if you're talking to a scientific community or a business community. So, I mean, it depends on the audience who's receiving that information, and if and therefore the quotes are sort of reprinted as you said it, and you mostly made sense if you're talking to another scientist, but it would mean a different thing completely to obviously people who are in the general public and are sort of have know someone who's suffering or whatever. And I get that. I suppose one thought I was thinking as well when you're both talking though, when you're talking about hype is, and I know you're both in sort of the academic field because you both work in universities, but is about funding as well. Because obviously a lot of hype around technology drives
yeah,
the funding, whether it's from an institutional point of view, but also from an industry and commercial point of view. And you only have to see currently right now if you're looking at biotechs and the drop in investor funding over the last 12 months in certain therapies because they haven't had the return as quickly as they thought there'd be, or there hasn't been as many approvals. So one thing I would ask you is though, is how important is it in terms of getting funding to have a certain level of hype to make sure that you're getting funding for the research that you're trying to do, or the work you're trying to do?
Yeah. So I think that if you're looking for, let's say, soft funding, like from a government grant or something like that, then you hope that you will have reviewers who are scientists, and of course you want to convey your project as clear and as exciting as you can. But I think that in addition to that, and like you say, biotech startups and stuff like that, when it comes to funding from investors and so forth, I think that's also a kind of a slippery slope. And I also kind of want to say the same about, media also, is that you want to be exciting. You want to show your excitement about your work. You have to do that, because if you're going to be just lukewarm about what you're doing, you're probably not going to get the grant. It's like, yeah, well,
And maybe you shouldn't be doing it either, to be
have
honest.
to show that you love it, right? And that you have, but I think that, sometimes when I've talked to, potential investors related to technology we're developing, it really happens fast. They say, "Oh my God, does this mean that everything is going to be fine in the world?" I mean, they can really hop to these incredible conclusions. And the same with also media, is that, of course, you want to tell about the cool stuff you're doing, but there's always this potential that they're going to overread and get you in a slippery slope of conversations. So, but I would say that my impression when it comes to funding is that a lot of funding agencies, at least in my impression, are still very interested in seeing work related to organoids, organ chip, and so forth. But I also think that to avoid this throw of disillusionment, as the Gardner hype cycle calls, we also have to make sure that we hold our horses a little bit, so that people don't think that this is going to be, like I said, Jurassic Park. We need to, make sure that has a certain level of soberness. So, yeah.
Mariana, what's your view on that, though? Particularly, obviously, you're working in a lab and doing a lot of research, so obviously, it has to be funded in terms of the work that you do.
Mm.
So, how does that impact you?
Well, so the funding agencies are, I think, still very focused on very defined projects. I think it's very, or at least my experience is that in order to get a grant funded, you really have to ask a very specific research question and really break it down into whatever aims that you think are going to answer your research question. I think research funding, except for funding agencies like ERCs, for example, there's very little space for big ideas and big out of the box thinking. And I think that's a shame, because that brings, that forces scientists to, in a way, focus their research on one little aspect of what is a bigger project. And but I understand also, of course, the concept of there is just so much money we can distribute to people. So there are several aspects of that, but I do think that the funding is a really big issue in terms of the science. And also, if I may, I would like to also say that I think it's very difficult for a scientist to do everything. Meaning we have to think of the science, we have to find the funding to do some scientists have to find fundings to pay their own jobs, right? We have to be grateful for being able to pay our own jobs, and it's a bit out of proportions in terms of what I think the potential that the scientific community has to actually what we actually do because of the limited funding, and also because, you know, we need personnel to do the work as well. So that means that you also have to pay people to do something, right? And then you also have to teach, and you have to disseminate, and you have to write articles, and you have to review papers, and you know, there's a lot of things we seem to have to be doing and be good at, and you know, it's like so it's I think you know, it's gonna keep us busy. I think, yeah, I think yeah, it's just unfortunate that you still see a lot of people dropping out of science because they just can't get their lab funded, and that's but that's I guess the unfortunate not necessarily natural selection, I think, of
Yeah, research, right. Well,
yeah.
look, we've been talking for 45 minutes. Can you believe that? So there was one more thing we're going to talk about. We'll maybe save that for the next episode. But one thing I do want to ask you is that sort of closing up. Um, is there anything in particular like that you'd like to talk to listeners about or highlight about organoids before we finish up today's conversation? Mariana, anything you'd like to say?
So for the organoids as in concept, as I think I mean, I think that organoids are certainly a way forward. I think we're not there yet. And I think that there's much research still that has to be done on normalizing also SOPs for standard operating procedures for organoids themselves. But I do think there's still a lot of confusion for a lot of aspects. And one of the things that eventually I think I would like to touch upon in our discussion is the ethics that goes around the study and the use of the organoids, and maybe even the concept of their commercialization in terms of pharma usage. But for some other time, I think.
That will be hopefully for a future episode.
Yeah.
And Steve, what about you then? Anything you'd like to
Yeah,
finish
no,
with the development?
I really have to second that. I think that um, the the focus on ethics and organoids, this also plays into a very exciting opportunity that organoids and organoid chip has, and that is again towards the multidisciplinaryism. And I think that even in natural science, we're seeing that, you know, chemists, you know, physicists, biologists, etc., are working together. But we can also see that this is a field that also the so-called softer sciences, like humanities, can also be actively engaged in and collaborating in, both when it comes to legal, ethical, even philosophical aspects. So I think that this is an excellent melting pot of science and knowledge. So I think that's going to continue to be the case.
Fantastic. And one other thing which struck me was that I know that both of you are traveling and attending various talks and events in the near future. So just in case any of the listeners happen to be at those events, so Mariana, where will you be heading to and doing some talks and listening to other talks around the topic?
So I will be at the end of September. I will be in Rome at the Organoid workshop, organized from Regina Celli, I think is the name of the institution, and I think it's a really nice chance to talk about the bladder projects that we have, and also a junior group leader from my group will also participate there. So Sofia Carcampuna, she will be there talking about prostate cancer, organoids, and the heterogeneity. So I think that would be a nice, that's going to be a nice meeting, and a very complimentary to the organoid meeting organized by Miles, I think.
Fantastic. And Steve, what about you?
So tomorrow I'm heading off to Tübingen. I'm going to be giving a talk in a workshop for the EUROC organ on a chip workshop that they have there, with hosted by, amongst others, Peter Loskill, who's one of the greats in the organ on chip work. And I'll be heading also to the Nordic Innovation Fair and the Nordic Life Science Days events in Copenhagen and Malmö in September. And then in October, I'm going to be going to the International Symposium for my favorite topic, chromatography, in Liverpool, hoping to see some of the Beatles sites and stuff like that. So there must be some stuff on that. So that'll be it for me. Yeah.
All right, fantastic. Well, look, listeners, if you happen to be at any of those events, make sure you do say hello to Mariana and Steve and tell them that you've listened to their podcast and tell them what you think. I'm sure you'll have loved it by then, though. So there you go. It's lovely to see you both. And one other event I should flag up, because obviously they're helping support this wonderful podcast, is make sure, if you haven't registered yet, to check out the Word Plus 2025 event, which is going to be near Cambridge in February. The link again will be above this podcast, so you can learn more about that. And it's all part of the WORC.Community website as well, so you can learn more there as well. So viewers or listeners, I keep saying viewers because I do a podcast, so I keep saying that, but listeners, I should say, make sure you like and subscribe this episode and subscribe for future episodes, and please let Mariana, Stephen and myself know if you've got any feedback, and also if there's any particular topics you'd like to learn more about or know more about that we can cover in future episodes of the podcast. So until next time, as always, I hope you stay well and stay safe. So it's goodbye from me, and it's goodbye from
Both of
Bye.
us. Bye bye.
they're waving, not realizing it's a podcast. I was going to see.
Oh, yeah, true. All right.
And goodbye for Mariana, and goodbye for Steve. Just in case we don't release this video. And it's goodbye from all of us. But as I say, stay well, stay safe, and hopefully we'll see you for the next podcast episode. Bye bye.
I'm waving again.
I'm waving. Bye bye.